Hey guys, welcome back to MedVidsMadeSimple. In this video, we're going to see about Staphylococcus. Now let's begin. Staphylococcus was first observed by von Recklinghausen in a pus sample. So Staphylococcus literally means bunch of grapes in Greek. Okay, they basically belongs to a group known as gram-positive cocci. Gram-positive cocci means purple-colored bacteria which are spherical in shape. They are identified using a technique known as gram stain. If you guys want me to make a video on gram stain, the techniques, principles, uses and all, please comment below and I'll make a video on that. So the gram-positive cocci are further classified into two families, family Microcapsule and family Streptococcus. The family Microcapsule includes four genus, starting from Micrococcus, Planococcus, Stomatococcus, and Staphylococcus. The other family is family Streptococcus, which includes Streptococcus, Pneumococcus, and it also included one more genus known as Enterotoxin. But now Enterococcus is placed in a separate family known as Enterococcus. We will not be discussing about Streptococcus in this video. So the first three genus which are mentioned here, the Micrococcus, Planococcus, and Stomatococcus, are usually not pathogenic to man. Okay. However, the fourth one, which is Staphylococcus, is the most pathogenic among all these organisms, which are all these genus which are listed here. How do you differentiate between Micrococcus and Streptococcus? It's very simple. You do a test known as Coagulase test. Micrococcus are Coagulase positive, which means all those four genus which I told earlier, such as Micrococcus, Stomatococcus, Planococcus, and Staphylococcus, are Coagulase positive, whereas Streptococcus bacteria are Coagulase negative. This is a basic and must know. Okay. Now, after identifying Staphylococcus, you can further classify Staphylococcus based on a test known as Coagulase test. This test is separate from the previous test which we saw earlier, which was Coagulase test. This is separate. Okay. Based on Coagulase test, you basically classify the Staphylococcus into Staphylococcus aureus, which is Coagulase positive, and Coagulase negative Staphylococcus, which are, as the name suggests, they are Coagulase test negative. This includes bacteria such as Staphylococcus epidermidis, Staphylococcus lugdunensis, etc. So how do you remember Coagulase test and Coagulase test? So according to alphabetical order, both starts from C. However, the second letter, consider the second letter. Coagulase has A, Coagulase has O in the second letter. Okay. So which comes first? Coagulase comes first. Which test do you do first? You first differentiate Micrococcus from Streptococcus. It's the first thing you do. So Coagulase comes first. So Coagulase comes second. What do you do second? You'll further differentiate Staphylococcus into Staphylococcus aureus and Coagulase negative Staphylococcus. So this is the second thing you're doing. So Coagulase test comes second. This is a very easy trick to remember which test is used for what. So these are the general features about Staphylococcus. They are gram-positive cocci, they are arranged in clusters, they are non-motile, they are non-sporing, they are facultative anaerobes, they are capsulated or non-capsulated. I'll explain all these terms. Gram-positive cocci means they are violet in color after a technique known as gram stain. Okay, so they are violet in color, and they are cocci because they appear spherical in shape. So that's why they are named as gram-positive cocci. Now they are arranged in clusters. It's very interesting. Do you know why they are arranged in clusters? This is because, okay, let's make this simple. Just close your eyes and imagine a cell dividing. Okay, consider one cell which is dividing into two daughter cells. What happens normally after cell division? Two daughter cells remain separate and they start dividing further. However, in Staphylococcus, what happens is they divide, but The daughter cells remain close together without getting separated. That's why they are arranged in clusters. And the cell division occurs in multiple planes, not only in single plane. These are the reasons why they are arranged in clusters. Simple, right? They are non-motile, which means they don't move around just so easily. And they are non-sporing, which means they don't produce any spores. They are facultative anaerobes. Now, what is facultative anaerobes? Facultative anaerobes means Staphylococcus are basically aerobes. They grow very well in the presence of air. But even in the absence of air, they can grow by altering their metabolic pathways. So that's what this term basically means. Facultative anaerobes means the bacteria are good aerobes, but they can also grow in the absence of air. Okay. Now, most of the Staphylococcus are non-capsulated, which means they are not surrounded by a capsule, but however few are surrounded by capsule. Okay. Now, Staphylococcus aureus. Staphylococcus literally means bunch of grapes, as I have told earlier. This is a Greek term. Why did they keep the name aureus? Basically, what happens is when the Staphylococcus aureus is grown on nutrient agar, golden yellow colored colonies are produced. So that's why golden yellow is named as aureus. So why is this golden yellow in color? It's because they produce a golden yellow pigment known as beta carotene, which gives the golden yellow color to those colonies. Now let's see about the virulence factors of Staphylococcus aureus. First, there are many virulence factors which are associated with the cell wall of the bacteria. This includes Peptidoglycan, Teichoic acid, cell surface adhesins, protein A, and Microcapsule. This bacteria also secretes many toxins such as Hemolysins, alpha, beta, gamma, and delta Hemolysins, leukocidin, exfoliative toxin, also known as epidermolytic toxin, Enterotoxin, and toxic shock syndrome toxin. They also secrete many extracellular enzymes known as Coagulase, Staphylokinase, H-stable thermonuclease, deoxyribonuclease, protease, hyaluronidase, lipase, and phospholipases. Now let's see about the individual virulence factors, the important facts you must know. First, peptidoglycan layer. You all must be knowing that peptidoglycan layer is the major component of cell wall in all bacteria. However, they are very important, especially in the cell wall of gram-positive bacteria. They are very thicker in gram-positive bacteria. Okay, this layer basically gives strength and rigidity to the bacterial cell wall, and they also have inflammatory activity, and they can induce inflammation. The next one is teichoic acid. This is basically composed of many polymers, and they help in the addition of the Staphylococcus bacteria to the mucosal layer. Okay, and they also prevent opsonization, which is swallowing by the protective mechanisms such as neutrophils and all that. They protect themselves from that. Now let's see about the cell surface adhesins. The first one is clumping factor. This clumping factor forms the basis for a test known as slide coagulase test, which is used in the diagnosis of Staphylococcus. However, slide coagulase test is not so reliable because about 15 to 20% of the bacteria shows slide coagulase test negative, and we can do one more test which is known as tube coagulase test, and that can be used to confirm it. So you need to know that the clumping factor is the basis for slide coagulase test. The other cell surface adhesin is fibronectin binding adhesin. Now protein A. The protein A has many protective mechanisms for the bacteria. This includes inhibition of opsonization. They are anti-complementary, which means they are against the complement system of the humans. So they also cause platelet damage. They mediate a reaction known as coagglutinating reaction, which is a very interesting reaction. However, I'm not going to mention this reaction in detail. I want to make a separate video on that, and when I make that, the link will be given in the description as soon as it is available. As I've told you earlier, few strains of Staphylococcus aureus have a capsule surrounding them, which is known as microcapsule. These are basically composed of polysaccharides. Okay, they're composed of polysaccharides, and they help in inhibiting phagocytosis. So it serves as a protective barrier for the bacteria. Now let's see about hemolysins. Hemolysins are very important toxins secreted by the bacteria. They basically damage the RBC membrane, the red blood cells membrane. They cause hemolysis, as the name suggests. They are classified into alpha, beta, gamma, and delta hemolysins. Here, the gamma and delta hemolysins are mainly associated with damaging human RBCs. The alpha and beta hemolysins do not cause damage to human RBCs. So remember, only the gamma and delta hemolysins can damage human RBCs and cause their lysis. So there's one more interesting fact. The gamma hemolysin acts together with one more toxin, which is known as leukocidin, to exert the hemolytic and leukocidal activity. The hemolysin will cause hemolytic activity, the leukocidin will cause a leukocidal activity. The next toxin is exfoliative toxin. The exfoliative toxin causes staphylococcal scarlet skin syndrome, abbreviated as SSSS. This is most common in children compared to adults, and this can cause wide variety of clinical manifestations, starting from mild blisters, bullae, to very severe features such as the one which had the Nikolsky's sign start to appear. What is Nikolsky's sign? Nikolsky's sign is when the epidermal layer of the skin can be peeled off the underlying skin surface. So this is somewhat severe. So this is the wide spectrum, which can be caused by wide spectrum of manifestations caused by exfoliative toxin. This toxin is also known as epidermolytic toxin. The next toxin is enterotoxin. Enterotoxin is responsible for the food poisoning caused by Staphylococcus aureus. This toxin is already preformed in the bacteria, so when you are consuming a food containing contaminated with Staphylococcus aureus, so if this toxin is already present in the food, this acts rapidly as soon as you consume it. But however, the symptoms start to resolve in short time, such as in about 8 to 10 hours. There is no specific treatment for enterotoxin. Just need to give supportive treatment such as correcting fluid and electrolyte imbalance. And the clinical features are as seen in any other food poisoning, such as vomiting, diarrhea. If these two are severe, this can lead to dehydration. If dehydration is severe, which can lead to hypotension. Okay, so rarely seizures can also occur, but that is very rare. Now, how this act? So the enterotoxin basically stimulates the vagal nerve. Okay, the vagus nerve. If you have seen my previous video, gastric secretions, I mentioned that the vagus nerve acts to parasympathetic nervous system, so that's parasympathetic nervous system, and I mentioned the actions of parasympathetic nervous system on the GI tract. So you can see that video. I've given the link of that video in the description below. And they also stimulate the vomiting centers in the brain, and they increase the intestinal peristalsis. By doing so, they increase diarrhea, vomiting, etc. As I've told you earlier, supportive treatment is the mainstay of treatment for food poisoning caused by Staphylococcus aureus. The next one is toxic shock syndrome toxin. This is quite severe condition. The risk factors for toxic shock syndrome toxin manifestations are poor menstrual hygiene, such as using tampons for a long period. And abscesses, osteomyelitis caused by Staphylococcus. This can serve as focus from which toxic shock syndrome toxin can be released into the blood. So toxic shock syndrome toxin enters the blood and they stimulate many T cells non-specifically to produce excessive cytokines, which are responsible for the manifestations. The manifestations are really similar to any other shock. Okay, so starts from fever, hypertension, vomiting, diarrhea, liver damage, renal damage, central nervous system damage, etc. So they cause multi-organ damage. So the treatment for toxic shock syndrome toxin is drugs such as clindamycin and some other drugs which can be given. However, supportive treatment is very required. Okay, now let's see about extracellular enzymes such as coagulase. This is a must-know topic. Okay, why? I'll tell you. This coagulase enzyme combines with a factor known as coagulase reacting factor, which is present in the blood. Okay, so these two combine, the coagulase and the coagulase reacting factor combine, and then these two together convert prothrombin to thrombin. As you all know, when thrombin is formed, thrombin will convert fibrinogen to fibrin, and this leads to clotting of blood. Fibrin clots will be formed, and this is the basis for a lab test known as tube coagulase test, which I have mentioned earlier in this video. So this tube coagulase test is very important to know. You have to know that coagulase enzyme forms the basis for tube coagulase test. This coagulase test is used to differentiate Staphylococcus aureus from coagulase negative Staphylococcus, which I already mentioned. So this slide is very important to know all the points which are mentioned in the slide. And as I've told you, there are other enzymes such as thermonucleases, deoxyribonuclease, staphylokinase, hyaluronidase, lipases. Thermonuclease and deoxyribonuclease are present only in Staphylococcus aureus, and they are absent in the coagulase negative Staphylococcus. The staphylokinase is used to lyse the fibrin clots in the blood, and it will help in spreading the infection to the whole body. Hyaluronidase is helpful in breaking down the connective tissue network and spreading the infection. Lipases also helps to break down of fat, and they also help in spreading the infection to various parts of the body. Now, the last part of today's topic is pathogenesis, of how the Staphylococcus aureus causes the infections. The first step is colonization. Staphylococcus aureus normally colonizes mainly in skin folds such as axilla region, perineal region, and they also stay in anterior nares part of the nose. So they enter the body through cuts or aberrations. If there are any cuts or aberrations, Staphylococcus aureus starts to enter the body through them. They invade using enzymes such as proteases, hyaluronidases, which are mentioned earlier. So they use these enzymes, they secrete these enzymes to invade the body. So they evade the host immunity by various defensive mechanisms, as I mentioned earlier, such as by producing various toxins, and they also have substances like protein A, microcapsule, etc., which are useful in defense mechanism against human immunity. And by doing so, they spread to various organs, and they cause various manifestations such as skin and soft tissue infections, respiratory tract infections, urinary tract infections, bone infections such as osteomyelitis, etc. And they have a wide variety of manifestations. In our upcoming videos, we'll be talking about the clinical features, laboratory diagnosis, treatment, and a very important topic known as drug resistance in Staphylococcus aureus. And stay tuned to get the videos. If you have any doubts to ask, you can donate us $75 or more through patreon.com/MedVidsMadeSimple. By doing so, you can gain access to Ask Your Doubts to us through an app known as Discord app, and then you can download your lecture slides. You can get your name on our videos as major contributors for 9 months. Hurry up, this feature is available only for 10 people. Thanks for watching. Please subscribe to our YouTube channel MedVidsMadeSimple. Like, share, and tell your friends to subscribe to our channel. Thank you.